So what I realized after I did all that stuff that I just mentioned, all the data mining aspect, oh, oops, sorry about that. Let me grab this, close that. Let me grab it. I did all the data mining aspects, so now I split into two different groups. So this is specifically for RNA with antisense capabilities. What I mean by antisense is this particular function, that this RNA is able to bind onto other RNAs. Like this is the messenger. So this RNA with the HFQ can bind onto it and prevent it from being transcribed. So there are two groups that I have here. One is, according to literature, not known yet if they're capable of binding onto the protein, and the other group is that are currently known. Okay, that are currently known. So the blue one over here is the one that's capable. The red one is the one that's not capable, and the y-axis is what I was talking about, like how deterministic, how much variability is there within each RNA sequences. Okay, so what you want to remember is lower it is, the more variable, the more deterministic, less variable, more deterministic. Okay, it's stable. Just think stable and then not stable. That's one way of thinking about it. So after I listed that, you now see like there's basically two different distributions over here. So and we were currently trying to figure out why this is happening. And one of my suspicion, it has to do with what I have to say, like RNA world. Okay, so how many people kind of heard of it? Know what RNA world? Doesn't know? Doesn't know RNA world? Okay, the RNA world basically is suggesting that we came from RNAs. Do you buy that?
You mean from like the ancient ancestors'
Yeah,
primordial soup?
Like okay, so after that, what was it, like big giant soup or something? You like cook it and then it spits out a bunch of like phosphate, hydrogen. I don't know. You like mix them together and out come like life. Something like that. And so this step in between, they kind of call it. So the hypothesis is first, before there's DNA, there's RNA. RNA can function by itself. There was an experiment. I cannot name the experiment, but I know it was on those scientific journals that they're able to create this molecule of RNA, self-replicate itself, self-sustain itself. In a sense, they create a virus or some form of a virus. And it's crazy. Does it have life? We don't know. It's just in terms of like perspective. So that's our guess. That's the guess that current theory. People are highly debating this. Like, is this how we came about? Like RNA first like kind of function and then eventually evolved into more highly organized groups and then more complicated groups. And now that they're like putting together these proteins, putting together these amino acids together to form proteins, and then I don't know. That was somewhat the theory behind it. So what I was trying to say over here is, like, in order for RNA to function by, so back in the day, RNA function could perform a function by itself, but it's just more physically stable to have some form, either like a non-metal, a metal or just some type of protection that can make the RNA more stable and more interactive. So what I think was happening within here is back in the day, these RNA can function by itself. And eventually, even though they do the same thing, they perform its antisense capabilities. But after a while, they evolved that, okay, maybe by hooking up with the protein, it might be better for us to function. So what do you guys think? Does it sound crazy to you? Or That's what I think, by the way. It's not. There's no. I don't have any basis or any literature that can support this. It's just an idea. Crazy talk. But I mean, anybody has some ideas, and just shoot it out. Okay, so this is something that I saw at, you guys know Lauren Williams? There was a tech professor that studies RNA, came here for a talk, and he showed this, and I was like, oh, that's pretty cool. Did you see it? Did you go to that? No? Okay, so basically what he said was, so we have, usually in bioinformatics, or he somewhat did bioinformatics, but in the bioinformatics, we're basically making a model of something. We observe something, we make a model of it, and then we generalize it and then make a prediction, right? So oftentimes our problems are very complicated, like this chicken. Okay? Just like this chicken, the chicken is very complicated. So you know what physicists like to model them as? Like a round square. Okay? So when I saw this, I was like, wow, that's very true. I mean, if you think about it, if you remember those like physics questions that you did in high school, say, so what would happen if you shoot like James off the mountain at this angle? And, I mean, that's basically what you're doing. You're modeling the objects as, like you're generalizing the object as some sort of a shape and some mass, right? But the reason why I put this up is I know we're attempting to generalize a lot of what we observe, but it's always, we need to kind of second thought, think like, are we actually doing it correctly? Okay, because this is kind of somewhat for myself too. Because the world is a lot more complicated. Like this chicken does not look like that. Come on, no way. I mean, maybe. So okay, so now I'm going to the next section of my presentation. This is something that we're doing recently. So, do you guys know WinGen? Like Dr. Zhao's group. So they have this problem, in which I thought was really interesting. Is they're trying to predict transcription start sites. Basically, trying to grab the promoter region, and everybody know what a promoter is, right? Transcription start site. So you guys, are you guys familiar with like within the transcription start site? Usually, when this location here is usually GC rich, right? This is just all the same. So here's an accumulation across multiple sequences, okay, multiple transcription start site that I know it's the transcription starts right in the middle, okay, which is why you see a central, like a peak over here, because there's accumulation across different GC content calculations with a window size of 150. But it's just basically I count number of GCs, dinucleotides, and then move it, and every single time I compute one value and so forth. It's very simple. Okay, so that's the idea. But now here's the interesting thing. So at our lab, we have this program. It's called CYK. Essentially, what you can think of it, or oh no, sorry. So back to the entropy thing. So I plotted the entropy, and it also looked like this. So there, I don't know if I'm making myself clear, but Dr. Zhao's problem was for us to predict this. Okay, so right now I'm just gathering different features that I thought might be feasible for us to use. Okay, so right now I saw that, oh great, oh, okay, there's a dip right there. And it might be because of the GC content. So that is the reason why. But I just thought it was a cool picture to show. So here's a wild thing. Here's the thing that we are currently trying to figure out. And if you guys can kind of help input this, I mean,